Hey everyone, welcome to my channel, and here I am with the structure of the coronavirus. In my last lecture I described the structure of the coronavirus, and I also said something about the classification of the virus. So here I am going to discuss the mechanism of action of the coronavirus, that is SARS-CoV-2. So this virus, after getting inside the cell, how does it work, what does it do? So this is the structure, as we already know that these are the S protein, the spike protein, these are the M proteins, E proteins are there, and inside it there is an RNA molecule which is the genetic material of this virus. So before we start the mechanism of action of this virus, you have to know some basic molecular biology, and after that you will be able to understand the mechanism of this virus. So here I have a diagram. So this is the structure of our cell, and here you can see this is the cell, this is the cell membrane having the lipid bilayer, and this inside there is a nucleus. The nucleus contains DNA. So the DNA contains the genetic material that the cell requires to make all the proteins and enzyme, and the DNA instruct the cell to form different types of protein. So how it does it? The DNA first make a copy of it in the form of mRNA, or we can simply say RNA. So this the DNA does with the help of transcription, and then the mRNA gets out of the cell, and here it gets modified having poly A tailing in the 3' end and 5' capping here. So this is the modification of the mRNA inside the cytoplasm, and then there are something called ribosome, that is the cell organelle present both in prokaryotic and eukaryotic. This ribosome having the bigger and the smaller subunit, they are also called as the protein factory of the cell. This ribosome bind to the 5' end of the RNA and then move toward it. What happens is that the ribosome will collect one by one amino acid present in the cytoplasm according to the codon, that is the information present in this mRNA that was given by the DNA, and in this process the tRNA helps, and of course rRNA is there which is already present inside the ribosome. So here the tRNA will pick up one by one amino acid according to the codon present in this ribosome, in this mRNA molecule, and accordingly one by one amino acid will make a protein, and that is the protein which is the cell which the cell requires, and this protein can be an enzyme, or this protein can be for the cell membrane or for the nuclear membrane, for anything it will be modified by the Golgi body. So this is what the molecular biology of a cell, and this is called the central dogma of molecular biology, that is from DNA to mRNA, that is the process called transcription, and from the mRNA a protein is formed, and that process is called translation. So this is the central dogma of molecular biology. So what does the coronavirus deal with this? So it basically hijacks the cell. How it does it? Let us discuss. So here is the structure of a cell. This is the nucleus, this is the cytoplasm, and this is the COVID, that is the SARS-CoV-2. So this SARS-CoV-2 has a receptor molecule, and these receptors are the S proteins, and like that our cell also have some receptor molecule, and there are some specific receptor molecule on the surface of our cell, especially on the surface of cell membrane of lung, artery, heart, kidney and intestine. That receptors are called ACE2 receptors. The full form is angiotensin converting enzyme 2. This angiotensin converting enzymes 2 are very specific for this S protein of this virus, and they can easily attach with this receptor protein, and by endocytosis the viral particle, that is the RNA, is just inserted inside the cell. So, after it just attach with the ACE2 protein, the structure actually becomes like this. This is what the structure becomes. Here you can see this is the viral particle and this is the coat of the virus and this is our cell. So, this is the cellular envelope and the viral envelope incorporate it with the cellular envelope, that is with the plasma membrane, and it inserts its RNA inside the cell. Now, what happens inside the cell? As the RNA get inside the cell, the interesting thing about this RNA is this RNA contains exactly the poly A tail and the 5' cap as like of our RNA. So, our cell gets hijacked. The ribosome will work for the viral RNA. So, as usually the ribosome molecule will come and it will just do the protein synthesis as it does with the cell of its own. So, now it will do the protein synthesis here. The translation process will go on, and now the ribosome molecule will be doing translation process with this viral mRNA or viral RNA, and then it will be forming a protein, that is the viral protein, and this protein is called RNA dependent RNA polymerase, and this is what you have to remember, the main important protein. This RNA dependent RNA polymerase is an enzyme that depends on RNA and that will polymer, that is RNA polymerase, that will form a new RNA. So, depending upon this already present RNA of this virus, this enzyme will make a new RNA. That means, this enzyme, after it is formed, this enzyme RDRP will come on this here and it will form a new RNA molecule. But mind it, this RNA is the positive sense RNA, but this RNA is the complementary of that RNA. That means, it is from 5' to 3' and it is just a negative sense RNA. But after this enzyme again come to this side and move on it, like it will again form the positive sense RNA here. So, 3', 5'. Like this, the viral enzyme will form various copy of the RNA molecule in the host cell. That means, it has hijacked the mechanism or the machinery of the cell, and the cell is giving all its system for the viral cause. Now, what is the important thing is that sometime this protein, this virus enzyme RNA dependent RNA polymerase will move from here and it will go to some extent only. That means, it will not form all the copy of the RNA, it will form some smaller copies of the RNA, that means, the RNA which will contain some limited amount of genetic material, and that is called subgenomic RNA. And this subgenomic RNA are sgRNA, and what do they do? Basically, these are the smaller RNA that the enzyme makes because it wants to form more proteins. Which are the proteins? S proteins, M proteins, E proteins. All these proteins are formed by this RNA, this subgenomic RNA, like S protein, M protein, E protein, because the virus has to make many copies of it. So, for making many copies, it requires the envelope protein, spike protein, membrane protein. So, all of these proteins are made by this subgenomic RNA after it does the translation process here also. So, all these RNA become busy in doing the translation, and they will form this S protein, M protein, E protein, and there are other total RNA for this virus. Now, all these proteins, S protein, M protein, E proteins, they are the membrane proteins, will incorporate together, taking this whole RNA inside it, and they are processed in ER and Golgi body, and ultimately a new virus particle having its own protein, lipid bilayer, its own RNA molecule will be released. Not only a single viral particle, there will be thousands of viral particles that will be released from this single cell after it infect the cell. So, thank you very much for listening to me. Thanks a lot.